So next experiment which we are going to do is to study the distribution of stomata on upper and lower surface of leaf and to calculate the stomatal index. Basically we don't do that index part, but still how to do it we will going to study it, and it is specifically there in the CBSE syllabus. So first of all, what is stomata? As you see here, the stomatas are the microscopic pores present in the epidermis of the leaves and the young shoots and of the plant body. They are mostly concerned with the exchange of gases and helps in the process of respiration as well as photosynthesis. The stomatas are guarded by the cells, they are known as guard cells and surrounded by the cells known as the subsidiary cells. The shape of the guard cells shows the variation in case of monocot leaf and dicot leaf. In case of dicot leaf, it is basically kidney shaped. In case of monocot leaf, they are dumbbell shaped. So, on the basis of the distribution of the stomata, as it is given here, they are of different types which we will study. Mostly the leaves, the guard cells surrounded are the specialized cells appearing in the different forms of the epidermal cells and known as subsidiary cells. The dicot plant causes stomata only on the lower surface. That is the most important point. That in case of dicot, it is present on the lower surface, whereas then in case of metrophytic plant, the stomatas are also present in the upper surface. And in case of monocot like grasses and cereals, they are present on both the surface. On the basis of the distribution of the stomata, there are different types. One is your mulberry type or apple type, where the stomatas are present only on the lower surface of the leaf, for example apple. Second is water lily type, stomata are present on the upper surface, water lily or mostly the floating plant body. Third, potato type, where mostly found in the dorsiventral leaf, where the stomatas are present in the both the surface, but mostly more amount is present in the lower surface as compared to the upper surface. Then oat type stomatas are equally or evenly distributed on both the surface, isobilateral leaf, that is your monocot. So what you are going to do, Mom will explain you the procedure that you will have to take the fresh leaf, then you will take out the epidermal peel, then after that you will study the upper and the lower surface of it, of the leaf, and then you will stain it and you will study the stomata. So after doing the slide, you will have to calculate that from total number of stomata present, and you will have to find out the stomatal index, that is number of stomata by number of stomata plus number of epidermal cells into 100. Basically, it is not being done in the class 2 level, but still this is the procedure which you are going to find it out. If you are going to do in the petunia plant, you will find it out that the number of stomata is more on the lower epidermis and only few stomata are present in the upper epidermis. But if you are going to take the lily one, then you will find it out, it is equal. So as we discussed in case of dicot, that is what you are finding it out, that is being given here in case of petunia, and in case of lily it is given equally. Some precautions while you are going to do it, while doing the experiment, the curling of the peel should be avoided. Always use brush to transfer the peel from the vasculars to the slide, and excess of glycerin should be removed. These are the precautions which you will take.
Students, and I am going to demonstrate that for the number of stomata or distribution of stomata in dicot and in monocot leaf. So dicot leaf, this is China rose. So you have to pluck a face leaf. Look here, this is the dorsal and this side is ventral. So upper surface and lower surface epidermis you have to take. So what you do, you have to keep in this position. That lower surface should be close to the slide and upper surface away from the slide. Keep like this way in order to take lower surface of the leaf. take a blade and scratch. Just keep on the table and scratch slowly or gently. So gradually we will get a translucent part. I got that translucent part that is content your epidermis. And keep on slide. Next, put a drop of glycerin. Then cover slip. This is for lower epidermis. Next, take another slide. Keep another link in opposite direction. And again scratch. So you have to scratch this side. Again you get a translucent upper epidermis. That also you have to take a small piece and put glycerin and cover slip. And both the slide you have to observe under compound microscope in 10x. And count number of stomata in upper and lower epidermis. So generally lower epidermis contain more number of stomata and large cell of stomata are kidney cell. But upper epidermis contain very less number of stomata or also totally stomata absent. So you have to compare both sides epidermis. Next, onion, this is called spring leaf. Take out their isobilaterally. Look here, the position is like that. Both sides get equal amount of sunlight. So distribution of stomata also equal on both sides. So take this leaf. This is also called spring leaf. This is the lower side and this is the upper side. Same way, also you have to take the peel. Put on a slide and scrap it. Okay, using blade you have to scrap and put upper and lower surface and compare both the slide to find out number of stomata. And here what will happen, you will get equal number of stomata on both sides and guard cells are dumbbell shape. So in this we have to compare both the slides upper and lower surface to find out distribution of stomata. So dipotely lower surface contain more number of stomata and upper surface least number of stomata. But monocots will get equal number of stomata because both the side because of isobilateral clip. So this is about stomata. Next is you have to take transverse section.